Muscular dystrophies are a group of genetic diseases that cause progressive muscle weakness and loss of muscle mass. The most common form is called Duchenne muscular dystrophy. It's caused by a faulty gene that is not working to properly produce an essential protein called dystrophin. Proteins are molecules that play roles in the structure and function of our body's tissues. Dystrophin is key for how our muscles work. Since the dystrophin gene is carried on the X chromosome, Duchenne affects primarily boys. This means that females can be carriers, typically without symptoms, but can then pass it along to children. Symptoms of Duchenne can start as early as age 3, affecting muscles that move the hips, thighs, and shoulders. Muscle weakness gets worse with age, also affecting the heart and respiratory muscles, leading to life-threatening complications, including disease of the heart muscle and breathing difficulties. There is no known cure for Duchenne muscular dystrophy. Gene therapy aims to be a one-time treatment that may be able to slow the progression of Duchenne by introducing functional genes into cells to change how they operate. For instance, the gene would provide instructions to produce dystrophin. However, the dystrophin gene is unique. Here to explain why is Dr. Jeffrey S. Chamberlain.
Our genes come in a wide range of sizes and functional regions. It just so happens that the gene that gives cells instructions to create dystrophin is the largest known gene in nature. It's too large to package into any of the current vectors used to deliver genes to cells. Therefore, a smaller version of the gene that we call micro or mini dystrophin have been developed to fit inside the vector to be able to provide a gene therapy for Duchenne muscular dystrophy.
The working genes are delivered into the cells using a vector, which is often derived from viruses. Don't worry, all the viral genes are removed, and the vector is modified to only deliver therapeutic genes. The goal of the micro or mini genes is to improve muscle strength and integrity. However, these effects of gene therapy are still being researched. Other therapeutic approaches aim to prevent muscle weakness using different muscle proteins to compensate for the loss of dystrophin. There are even gene editing approaches being studied that would replace the dysfunctional gene within the muscle cells. Gene editing uses precise technology to make changes directly inside the cell. With any of these approaches, increasing functional dystrophin or other regulatory proteins can result in improved muscle function. Researchers continue to work optimistically on gene therapies in clinical trials until they are safe and effective. For more information and resources about gene therapy for Duchenne muscular dystrophy, visit asgct.org.